Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing another really fascinating guest for you involved in science, technology, medicine, creating a better tomorrow for many people out there. Today on the show, we are honored to be joined by Dr. Dragan Primorac, who is a globally recognized physician scientist whose work spans domains from personalized medicine to regenerative therapies to forensic genetics. From 2003 to 2009, Dr. Primorac served as the Minister of Science, Education, and Sports for the Republic of Croatia, and that is the ministry and the government that's in charge of primary, secondary, and tertiary education, as well as the country's various research institutions and sport. Dr. Primorac is also the founder of the St. Catherine Specialty Hospital in Zagreb, which is the official hospital of both the Croatian Olympic Committee as well as the official hospital of the Croatian Football Federation, and is affiliated with multiple medical schools and the Ministry of Science and Education. It was recently became a scientific center of excellence for personalized medicine in Croatia. Dr. Primorac is also the co-founder of the International Society for Applied Biological Sciences. They're a global scientific platform organized with the Mayo Clinic that brings together various Nobel laureates and researchers from all over the world. He is also a pioneer in the area of DNA technology to identify war victims and forensic genetics. We'll be getting into that a lot on the show. Extensively published with over 400 scientific papers, multiple international honors. He also serves as a professor down the road here at Penn State, as well as University of New Haven and several European medical schools. Also a major Taekwondo expert and inductee of the Taekwondo Hall of Fame here in the U.S. Did both his medical degree and PhD at University of Zagreb School of Medicine, went on to do postdoc work in molecular medicine at University of Connecticut Health Center. And we're honored to have him with us. A lot of really cool topics to get into today. Dr. Dragan Primorac, thank you so much for taking time on the show today.
Thank you, dear Ira, for having me with you. And it's indeed, it looks like a very complex life, but as my dear friend, Nobel Prize laureate, Shimon Peres said once, no room for small dream. So that's exactly what I'm doing.
Absolutely. Absolutely. Yeah. And let's start off. Let's go back a couple of decades, because as with everyone that comes on the show, I usually take a look at their PhD, and I took a look at yours, which you had to do with the molecular biology of this disease, osteogenesis imperfecta, which just a year or so, I think, after your PhD, I think the world learned about this disease from the Bruce Willis Samuel Jackson movie Invincible. The bad guy had this genetic disorder. I would love to hear a little bit about how you got started at the beginning, but also sort of what brought this to your doctoral thesis and why you were interested in it to begin with.
Yeah, thank you. As a young physician, I never accepted the philosophy that we are not able to do more than what we have in the medical books. And I was selected among a few physicians to come and to start my postdoctoral fellowship. And I was very fortunate to come to University of Connecticut Health Center in Farmington, Connecticut, and start working with David Rowe, who is an outstanding man, and I would say not only great pediatrician, but great human being. So at beginning, indeed, it was just beginning of the new area of the PCR enter our laboratories. So we are trying to see what we can do for those kids, and I was deeply emotionally touched because I met so many of them. And, on the early 90s, I learned different types of osteogenesis imperfecta, or brittle bone disease, what we call. A certain number of the kids are having fractures, some of them having more than 50, 60 fractures till the puberty. Unfortunately, some of these kids die because of the osteogenesis imperfecta type 2. So, it was a great interest in both in Europe, United States, America, try to understand the mechanism of the disease. As a young physician, I have great enthusiasm, probably, on the level that a few times David told me, Dragan, I think you should, go home much earlier than what you're doing, because I was just combining, morning, nights and everything, because I was really, really hoping we are going to have discovery which is going to help eventually kids, not only for the diagnostics, but the treatment. And, during that period, I was very, very, very fortunate and lucky to meet outstanding physicians, scientists. And for the first time in my life, I learned why it's important to work on interdisciplinarity, because we have molecular biologists, we have biochemists, we have pediatricians, we have radiologists, we have trauma surgeon, orthopedic surgeon, and so on. So each of us try to put a puzzle in order to understand the mechanism of this disease. And during that time, I remember for me was very, very, very challenging to play with the cells and to grow the cells we call fibroblasts. Because, once in a while, for the reason that we are not able to understand, those cells get infected. And the problem is you spend 2, 3 months, 4 months, and everything what you have done, it's gone. So it's happened with me a few times at the beginning, but even motivated me more to do. And what also very important to say, everyone was so generous, because I was young physician entering into molecular biology, and the level of the patient passion and love that I witnessed, it's really helped me that I transfer that technology for the young generation in the future. And indeed, I will just tell you briefly on the morning that I remember that I was able with my team to find the particular mutation on the region we call intron, at the beginning of intron, that was reason why the entire messenger RNA that belongs actually to DNA that should go on the cytoplasmic compartment and give a protein get stuck in nucleus. So when I saw that, I said, wow, we are having something completely new. One small letter at beginning on the portion of the gene which anyhow should not be translated to the protein, it's causing such incredible problem. So I remember I was running to the David and Mary Louise Stover, that she was a technician. And David said, let's go repeat it. And we repeated a few times. And I would say a few months after that, we have in our hands the new mechanism of that particular model of osteogenesis imperfecta, why this messenger RNA, which is eventually important to leave nuclear compartment and give the protein, why is missing? What is wrong? And few months after that, we went to a big meeting organized by American Society of Bone Mineral Research, and I was fortunate to receive Young Investigator Award, which make me, incredibly, incredibly happy because somebody recognized, my work. And that's for the first time where I understood in the medicine that it's better to have 50% of the fully functional protein, which, give you disease indeed, but a mild type, rather than have 100% of the protein, which 50% is a damage and 50 is normal, that you are giving severe or lethal osteogenesis imperfecta. And that was the moment that entirely changed my life. And since that, I'm deeply in science. I appreciate science. And to be honest with you, I cannot imagine myself as a physician living one single day if I'm not in laboratory and if I'm not working with the science.
Absolutely. Yeah, it's great that again, and looking through your extensive bibliography here that you've stayed with it. It wasn't, just one discovery that you made back in 1997. And we'll come back to that when we get to some of the discussion about personalized medicine and regenerative medicine and so forth. But I want to keep going, along sort of the path that you followed after this, because as you mentioned, you're doing this amazing postdoc work. You're at University of Connecticut, but you're also, simultaneously training extensively in forensic science at this point. And you're everywhere, from the Connecticut State Police Forensic Science Laboratory. You're down in at Walter Reed at then the Armed Forces Institute of Pathology. And sort of this leads into your role, and you co-found the Laboratory for Clinical and Forensic Genetics, in Croatia, and becoming this longtime member of this office for detained and missing persons per the war. I would love if you could talk a little bit about this role, a little bit of how this all emerged. I guess obviously following the war, because we actually talked a teeny bit a few months ago. Here in the United States, you're aware of this. Probably a lot of the audience is not, that we in the U.S. have a national missing and unidentified person system with something like the remains of 50 or 70,000 folks that we just don't know who they are, and ultimately identifying them is a major task. But now we have tools in front of us. Take us back a little bit to when you initially took on this other role, because it's the 90s still, tools still being developed, but a very important responsibility for you.
Thank you. Actually, it was very, very painful period of my life, because I was working as a young postdoctoral fellow, just get incredible results on osteogenesis imperfecta. I was also doing a lot of work on the osteochondrodysplasia.
Yeah.
The cartilage since 1992 was my love, and it's still today. When I do a lot of work with the world leading athletes, they ask me, how come that you are so deeply involved in cartilage? I said, it's coming from the 1992, when I was doing a lot of research on the chicken, actually on one model called nanomelia, in order to understand the mechanism of this disease and why we are not having the certain protoglycans in the cartilage. So one of those days when I was in the lab, I received a call from Croatia. And it was a very disturbing call. And a colleague and friend of mine told me, we're having incredible difficult situation, because we just discovered one mass grave. And we don't know who is in the grave, because we don't have technology. And by standard forensic methodology, we are not able to identify those people. And I said, how I can help you? Because, I was really not involved in the forensics till that time. Even, my thesis I did on the Department of Pathology. So he told me, we just realized that FBI, United States Army, is entering on the completely new area, which is forensic genetics. And you may, jump in and start, learning about new methodology, and eventually it can help us. So, I made few calls, and you are completely right. I made call to the Armed Forces Institute of Pathology. It was outstanding man, Professor Mitch Holland, who was working on the Tsar Nikolaus case in Russia. And I also call FBI, where, very famous Professor Bruce Badoli was working, who was the guy who was actually did remarkable work in the field of the forensic genetics. And I make quite few calls, including call in Connecticut State Police, where Professor Henry Lee, who was chief of the forensic laboratory, then in New York, Michael Baden, legendary, who did autopsy of President Kennedy, Martin Luther King. And to be honest with you, that was one institution, one organization that was facilitating all of that. It was AmeriCares. AmeriCares from Connecticut, huge charity organization. And that's how I met Stephen Skakel, actually, who is related to the Kennedy family. Soon after that, it was 1993, I met President Bush and his wife, Barbara. And since the first moment, she realized how important this project is, and she helped me tremendously. So from 1993 till she passed away, we've been in touch. Even when her son, George Bush Jr. came to Croatia to visit, I was Minister of Science, Education, and Sport, so he came with a message from his mother. So there's a lot of emotion. But Ira, at the beginning, I was really involved in the pioneering work on forensic genetics, because at best of my knowledge, And same, has been said by others. Nobody did it. Identification of skeletal remains on such huge, large scale from the mass grave. And at beginning, indeed, we have to start working on the different protocols. You have to start working on different methodology. We didn't have any clue why we are not getting DNA from certain, bones, why we are getting from other locations. So during that time, we start learning about bacterial contamination, contamination with the heavy metals, and so on. And with tremendous help from Connecticut, New York State Police, Analytical Genetic Testing Center. It's an outstanding man. He was a very close friend of mine, Professor Moses Shanfield. Unfortunately, he passed away. He was one of the greatest geneticists in the United States of America. And as I said, Bruce Badoli, Mitch Holland, and many, many others. Basically, we start building completely new philosophy. And we start collecting blood samples from the living family members who are missing somebody, kids, parents, grandparents. And then we went on the site and we start excavating remains and getting every single bone, sending to Department of the Forensic Pathology, trying to get rid of the all contaminants, extracting DNA and start amplifying. And it was very, very early phase where the probability of identifying somebody was about 60 to 70%. Today we are talking 1 per billion. And indeed, it was huge responsibility, but also emotionally, it was very, very difficult. You can imagine, few times we've been flying back to Croatia during the O.J. Simpson trial. So even all media in the United States be asking me or Michael Baden or Henry Lee, what is more important in the war than O.J. Simpson that you guys leaving states and going to Croatia? But I have to tell you, it was incredible humanitarian effort that was bridging science and the justice. Because, the message, beside identifying human remains was that we are going to find the perpetrator, that we are going to find the people who committed the crime, because everyone has to be responsible for that. Otherwise, the question is, which kind of the human beings are we? And when we published the paper, in the most prestigious forensic journal, Journal of Forensic Science, which belongs to American Academy of Forensic Science, it was actually, best of my knowledge, the first time that one group of the scientists published data using DNA technology from the mass grave. And since that, we are deeply involved in forensic science. And once again, I would like to thank all my friends from the United States helping me. And since that, we are friends. They are coming all the time. And the same kind of the philosophy we used after horrible terroristic attack to twins and any place in the world, same principle, same philosophy. And emotionally, it was very, very difficult to talk with the family members. It was a lot of media. And then eventually we came to the point that analysis of the genomic DNA, which we are getting 50% from our parents, was not sufficient. And we moved to mitochondrial DNA, which we are actually inherited from our mother and having much more copies. We are talking about thousands and thousands of copies. So in most difficult cases where we are not able to identify human remains, we've been switching from genomic to the mitochondrial DNA.
Sure.
But that's one additional thing. I didn't believe that on the 2007 additional big step forward we made, actually collaboration of the Bosnian and Slovenian scientists, one of the first identification of civilian victims from the Second World War, and we showed there is a possible. And I said, okay, guys, you have to take care of the political issue because it's not easy to enter, other country and start digging the graves. But, my mission is to show you that science can do it. And, a few years after that, other big case came, with identification of the sister Maria Crucifixa Kozolic, that she died on the 1922. And actually 100 years after her burial, the church decided to start a process of beatification, basically nearly a century after the death. So we still use same philosophy, and still the same partners from the Penn State University, Mitch Holland, and group from the Armed Forces Institute of Pathology. And kinship analysis confirmed actually there is a 500 times higher chance and likelihood that the remains belong to the second-degree relatives of the living niece of the Sister Mary. So the power of the forensic genetics, it's incredible. And it makes me very happy because I was lucky and I have fortune to be involved since beginning.
Yeah, it's again an amazing part of your story, and again talking about, as you mentioned, the interdisciplinary nature of something like forensic genetics and bringing together the right pieces, and at the same time, as you're mentioning, how technology is constantly evolving and the ability to adapt and improve along the way. But again, it's another very impressive piece of the story. Before we get to what you're doing currently, which is a lot. I obviously want to start one other place, and that is obviously your tenure as Minister of Science, Education, and Sport, 2003 2009. And here again, a long list. You're involved in everything from reorganizing research organizations in Croatia, standardizing the bachelor, master, doctoral systems with across Europe, creating new systems and agencies for quality assurance, accreditation. But at the end of the day, I think a lot of it comes down to one major thing, a major proponent of science education and sport, but also public investment and increasing that investment in these incredibly important parts of our society. I know it's odd because in 2025, we're talking a lot about defunding science, which is scary. But again, take us to 2003, 2009, a very important part of your career journey.
Thank you. As a scientist, I received a call from the Croatian Prime Minister who called me and he said, please help us. You have experience both in the States, in the Europe, some other countries, and immediately I told him, I would like to transform Croatian education system and science as Croatia is a knowledge-based society. And I told him, in my opinion, the biggest risk is not taking any risk. So I'm going to do something completely different than anyone involved in politics. I'm going to risk, but I'm going to do my best actually to transform Croatia. So I remember those days, which in the period of the 6 years, basically, I was able to double the budget from 0.9 billion euros to almost 1.7 billion. And you can imagine, Ira, that I was able to convince the prime minister and the government to introduce in Croatian primary school mandatory learning of the 2 foreign languages. So Croatian plus 2 foreign languages. In such a short period at the time, we were able to integrate information technology in every level of education system. All Croatian universities, research institutions, all schools, they be connected with high-speed internet. And it was early, like 20, it was like period when everyone was discussing in which direction to go. Then I was able to secure the budget to providing free textbook, transportation, and the students' housing, making education more accessible for everyone. I strongly believe that one project that is actually, I would say, so important for any country, it's a project which I call from the brain drain to the brain gain to make it functional. So in the period of 6 years, we were able to bring about 150 top PhDs and top scientists from Australia, Canada, from United States, Germany, back to Croatia. And those people, they changed the mentality of the environment. They brought a project, knowledge. In such a short period of time, 10,000 new jobs in education sector I was able to open. And you will be amazed. I mobilized entire country to build 350,000 square meters of space at Croatian universities. We opened 3 new universities and 6 polytechnics. I said, that's not all. Croatia does not have a future if you're not going to increase number of the students. So in such a short period of time, we were able to increase number of the students for about 40,000. And in the same time, we were able to build 350 schools. And what made me incredibly happy that at one moment, 2010, it was Newsweek, They publish huge article, the world's best countries in the health, education, economy, politics, and quality of the life. And the Newsweek in 2010 placed Croatia on the 22nd place in education, ahead of the countries like United States, Israel, Greece, Portugal, Spain, and so many. Actually, Croatia was ahead of the 12 countries that belong to the G20 group, which makes about 85% of the world capital. And my message was very strong. If you believe that is possible, nobody can stop it. And I said to everyone during that time in the media, I said, excellence is commitment. It's not one event. And I was telling to the all people in Croatia, the best way to predict our future is to create it. Let's guys do it together. And I remember when I was invited by Science magazine, when the David Baltimore, still Nobel Prize laureate, he was the one who was heading the big organization. They called me basically to present the model. How is that possible? How you're able to transform small Croatia is in a knowledge-based society. You know, and I remember that I was giving lecture, and when people told me, were you scared to see? Because, you know, once in a while you see that politicians are replaced if they're not listening, somebody who is in charge. And I said, don't be scared. A scared man is annoyed by every noise. You should do what you believe that is best for your country. So during that period, you know, we established tremendous relationship with Japan, with India, with NIH, with Israel. I was visiting a few times, chief scientist officer, and actually took quite few outstanding models, especially on translational medicine and on the entire concept of the patenting results from the research, where I found Europe to be very, very, very weak.
Yeah, it's again,
it's amazing. You know, I'm still thinking how I was able to survive politically that period, because it's an entire revolution. We were building Croatia from the kindergarten to the PhD program, and it was only one message. Only one message. You know, I was telling everyone, please remember, excellence is commitment, not one event.
Excellent point. Really excellent point. And it's something we should, again, it's something we should hopefully be hearing more about in 2025. But that's a whole other story. But again, very extremely impressive. Let's transition to everything you have going on now, because as mentioned, that you have quite a lot. I thought we could take some time to drill down on some of the current projects, because they're just so relevant to a lot of the themes that we touch on in the show. And I think if we could look at sort of one basket first, sort of your initiatives in personalized and precision medicine and genomics. Would love to hear about a couple of these projects. One that obviously stood out just because oncology is a hot topic for us, AI, of course. Would love to hear about your work on a program known as OncoOrigin AI, which is focused on a really interesting concept, sort of the cancer of unknown primary source, which we don't get into too much, but is a really interesting concept that says, hey, in these advanced patients, the cancer cells that we're looking at may have started and reprogrammed and changed a lot from where they originally started. This leads to a lot of important information on prognosis and ultimately the resolution of these cases, how we treat them. Talk about OncoOrigin AI a bit, because I think it's extremely important and timely.
Thank you. Just 5 months ago, in a partnership, one of the leading academic healthcare organizations in the United States of America, University of Pittsburgh Medical Center, in partnership, we open new clinic in Croatia. So idea is that such outstanding center with very comprehensive approach to the cancer care, together with my team, provide everything basically from the genomic, or what we call multi-omics, including proteomics, analysis of transcriptomics, epigenomics, lipidomics, and so on, together with best possible diagnostics and the treatment. and we've been concerned because, as you know, the targeted therapy is becoming priority. Even, it's so striking. Actually, the fact that Food and Drug Administration approved, I think, 198 cancer drug between 1998 and in 2022, which is 43% of those based on specifically molecular tumor profiling.
Yeah.
Which is changing everything. And then you are coming to the point that cancer of unknown primary represents a significant diagnostic and therapeutic challenge because it's approximately between 5 and 10% of the all malignant disease, and typically exhibits very high biological aggressiveness and rapid progression, and resulting in extremely poor prognosis. So that was really big challenge for me and my group because, it's incredible, but about 84% of the patient die within first year of diagnosis.
Yeah.
So we did a few studies on artificial intelligence and machine learning models where scientists from St. Catherine Hospital and US Dartmouth Health try to analyze over 20,000 different samples of metastases of the different kinds of tumors, as well as the mutations in more than 600 different genes. And actually, all results that we show, it's based on computer simulation. And the results are amazed. And if you're looking what we call receiver operating characteristics, area under the curve, we have score 0.97, and you can imagine that value 1 represents the perfect model that correctly classify every tumor type without any error. So we came so close. And indeed, we published that model. It's helping us tremendously right now, not on scientific base, but on routine clinical practice. Our idea is to increase number of the genes. And, in Croatia, since September last year, St. Catherine Hospital were able to provide the technology where we are doing together with the International Center for Applied Biological Sciences, all genome sequencing for every single patient that is entering hospital. So basically, we are creating huge DNA database, which is going to help the concept that we have in our onco region. But let me tell you also what we are doing simultaneously. It's a huge challenge right now for not only patient, which you correctly said, cancer of unknown primary, but for all others. We recently introduced completely new concept, which is much needed, called liquid biopsy, which is extremely, extremely sensitive, where we can obtain the results for basically 5 to 10 nanograms of what we call circulating tumor DNA, which represents only 0.1 to 1% of the total circulating DNA in the blood. So with all what I just said, we are just approaching to the certain goal that I decided to approach many years ago, but with the help of the UPMC and some leading institutions, including Dartmouth University, we are doing a lot. But let me just tell you one thing. If you see how complex human being is, just tell you that each of us are having about 30 trillion of the cells, and that about 1% of the cells being replaced daily. We understand right now that we need about 100 days to restore ourselves and to become equivalent a new you or new me, which is so critical in order to understand the completely new concept of regenerative medicine, which we are combining all the time and keeping close to the cancer. At the same time, we've been studying a lot of epigenetics of the cancer, which can be tremendously influenced by bacteria. But in the same time, Ira, we are having 30 trillion of the cells, humans, but 38 trillion of bacteria. And the question is, are we more bacteria than humans? And at the same time, if we are moving a little bit further, DNA is my love and passion. And if you check DNA in one cell, it's about 2 meters long. But if you're going to check entire DNA in the 30 trillion of the cells, it's actually twice of the solar diameter of the solar system, which is amazing. And the question is, how is possible that our DNA, which is having so many mutations, and we know by the different publications, indeed, it's very difficult to measure, that individual cell can suffer up to 1 million DNA changes per day. And I've been discussing about that with a great friend, Nobel Prize laureate Paul Modrich from the Duke, who actually found the mechanism of DNA repair. So there's so many mysteries that we are trying to apply, indeed, the best of our knowledge, when we are creating artificial intelligence program, as you mentioned, on co-region, because it's not anymore enough that we just play with the DNA. Right now, we are trying to understand more what we call multi-omics from every single aspect of the tumorigenesis, including epigenetics. And there's nobody on the planet who has a capacity to understand that without collaboration. So I think we have all be humble. We have to exchange our knowledge, and we have to have what Shimon Peres said once, all time in our mind, no room for a small dream. Because as a physician, I'm motivated to help my patient. And there's no money in the world, there's nothing in the world that can replace the feeling when I see the happy face of the patient that I was able to help, either with the progress on the diagnosis that was not possible just recently, either by introducing pharmacogenomics, where we are able right now to cut adverse drug reaction, either by applying mesenchymal stromal stem cells in treating patients with osteoarthritis, or recently pure MSCs to help patients with the COVID-19 and so on. So my nature is very simple. Books are great, but we have to think much above current knowledge. We really have to be surrounded with the challenges. We have to live to make a difference. And to be honest with you, as Gandhi said once, you must be change you would like to see in the world. Then you are witnessing why everything what we are doing making sense for me as a physician and for me as a scientist. And I'm never going to change that philosophy, because I think simply it's a privilege to be a physician, and it's a privilege to help others.
And speaking about challenges, and I just want to stay in the precision medicine world for one more moment with you, and we'll move on to some of the other really cool topics. But I couldn't help in the multi-omics basket. The one omics that to me is the scariest, because it's the most complex, is of course the glycome. And I couldn't help but notice that you published a lot with Dr. Gordon Lauk, who we had on the show a couple of years ago, most recently in the Croatian Medical Journal, on the role of glycans in personalization and preventative health care. This is an area, when I say it's scary, I know over the years when I hear people talk about the glycome and this whole area of biology, which is, in our 30 trillion cells, is involved in a lot of the cell communication and all the other things that the epigenome and the genome, when they transfer their information, it's got to go down these interesting pathways. Can you just say a couple words about sort of where we are with glycomics in the multi-omics basket? Because it's a tough one, but needless to say, you're working with the one of the best minds in the business with Gordon.
Yeah. So actually, what makes me very happy that in every single study that we are doing right now, we are trying to incorporate together with the genomics, multi-omics as much as possible. And I'll tell you a few examples. For example, quite a few years ago, in one paper, Gordon, myself, and our group, we've been able to show that changes in the glycans are visible 10 years before the patient developed clinical picture of the rheumatoid arthritis. So that's amazing.
Yeah.
So we're talking about prediction. We are talking about biomarkers. Right now, every single study that we are doing, we are incorporating glycans together with whole genome sequencing, with transcriptome and proteomics. But there is one issue which I would like to underline. There's a huge program, I would say one of the biggest projects in the EU on the osteoarthritis. We'll be using what we call microfragmented fat tissue, having a lot of mesenchymal stem cells, but other cells, including pericytes, endothelial progenitor, and so on. And in many studies, including ours, we show there is a huge potential anti-inflammatory, immunomodulatory, angiogenic, antimitogenic, and so on. And we did a huge study on the patient on the osteoarthritis, applying directly to the knee, microfragmented fat tissue, and expecting to see the changes in synovial fluid and the blood of the patient. Because what we witness, it's amazing. First of all, we witness changes on the glycosaminoglycans in the cartilage.
Mm-hmm.
We witness that patients are doing much better according to the all orthopedic scores. But we wanted to understand what is really happening. So by using multiomics and measuring time 0 before it is started, 3, 6, and 12 months, we get so many data from multiomics that even year after finishing the project, and by using superpower, I would say computer, and using artificial intelligence, we are still not able to analyze those data. And that's a huge challenge that we are witnessing today. And to be honest with you, when we are talking about multiomics, every day we are facing new challenges. Recently, we have a study that we show on a small cohort changing on the glycans after chemotherapy. We still don't know, is it good or a bad sign? So we have to increase the cohort. But I think by using the concept of the multiomics, I think we're coming to the position that we are going to get answers that we are never able to get earlier. And as I mentioned earlier, I just said a few words about pharmacogenomics.
Yep.
Which you already know that JAMA published paper many years ago, confirming that about 106,000 people only in the United States dying because of adverse drug reaction. And we are focused only on the certain gene variation important either for transport of the drug or metabolism of the drug. You can imagine what will be happening when we are including all these omic possibilities, including glycans, to get the right answer. But in the meantime, we have to do what we can to do. Certainly, pharmacogenomics is changing everything what we know about drug treatment. And today, every single patient that is entering St. Catherine Hospital is having possibility to do pharmacogenomics in order to avoid adverse drug reaction, which is, as I said, killing only in the United States more than 100,000 people annually. Or there's another example, considering that about 60% of the 700,000 people who die because of the suicide every year worldwide having depression.
Yep.
We have to do pharmacogenomics because only 30% of those patients reacting on the drug treatment. And without pharmacogenomics, you are really not able to understand and you're not able to prescribe best possible drug for that patient. And please remember, out of the 700,000 people who are committing suicide, it's about 60% of the patient with diagnosis of depression of different bipolar disorders.
That's no, a staggering figure. But no, I appreciate you connecting these two worlds in terms of the pharmacogenomics and I used to the area of neurological disorders, mental disorders is just the beginning in terms of applying these technologies. But I'm kind of interested in bring it back. I mean, you talked a lot about obviously your work in tissue engineering and specifically cartilage repair, which obviously you talk to anybody in the field. This is it's tougher than regenerating parts of the brain, and I mean it's one of the holy grails out there. And you've been as you were explaining, and there's a recent paper on terms of tissue engineering and future directions was out in 2024, and again you were talking about, as you just did, the unique cell types, the scaffolds, the interesting sort of hybrid decellularized extracellular matrices and all that. And it's a fascinating paper. We'll put a link to this as well. But later on in this review, you go into sort of the future potential of combining the gene therapies and the gene editing techniques and sort of the newer technologies that are coming down the path. I sort of like it to take us a little into the future with this one, because obviously it's an area that was kind of recalcitrant, let's say, to traditional regenerative medicine techniques. We're in a new era now, and we'll love to hear about sort of your future vision for these technologies. And then obviously this feeds back to your work on osteogenesis imperfecta and sort of a lot of the monogenic diseases, but where you see the CRISPRs and sort of these newer techniques in terms of gene editing fitting into the overall approach, because multidisciplinary, but it's all here now. So
yeah. Really, to go deep in this, it helped me a lot from my education on the forensics, molecular biology, pediatrics, personalized medicine, because it's very, very difficult to follow all of this if you're really not connected with the top people from the field. Because as you said, only if you are discussing about application of the microfragmented fat tissue, actually, which changed my life internally, because when we start doing intra-articular injection of microfragmented fat tissue, having all these cells, including mesenchymal stem cells, pericytes, and endothelial progenitor cells, we witness significant increase of the glycosaminoglycans content in the cartilage. Actually, according to the literature, for the first time. So the patient reported reduced pain, improved joint function, and we witness results for 24 months. And right now, after 3 years, patient coming back for the second procedure and so on. During the time, I start having collaboration with few different groups. One group from the United States of America, and there is Professor Antonio Talla, who is doing a remarkable job on the Wake Forest University on the tissue engineering and organ production. A second group in Israel, Bonus Bio Group, doing tremendous work on actually by using the same concept technology that you mentioned, bone production. And actually they produce the bone by using MSCs on a scaffold. And actually this bone which they produce, they implemented in the patient who is functioning completely normal, which is revolution, in my opinion.
Mm-hmm.
And actually, they came to Croatia a few times. I was with there. We have a very, very strong contract of collaboration and so on. We became later on the friends and thinking about progress in the future, what we're going to do. And as a matter of fact, as we speak, recently, Professor Tejas Patel from India, one of the leading cardiologists from India, came to Croatia. And right now, the group from the United States, India, Croatia, and Israel, we are having consortium where we are starting, hopefully by the end of the year, clinical trial in applying directly via catheters to the left coronary artery on the patient with inoperable coronary artery disease in the India, which is amazing. So we've been preparing this clinical trial for more than 2 years. 4 different countries. We put it all our knowledge together. And as a matter of fact, when the Indian Prime Minister visited Croatia recently, one of his first question to me was, Dragan, when we are starting the clinical trial? So it make me incredible, happy and proud, because you are having the number one man in the country giving such incredible support to the scientific project. So those are things right now which helping us to understand how we can go further. And indeed, as you speak about more complex organs, situation, it's even more complicated because right now
Mm-hmm.
what we understand more about bridging with the blood vessels. We understand more about different scaffolds. We understand more about potential of MSCs. And still quite few things we don't understand. And I just gave a lecture in the Padova in Italy where we are discussing how to do a tissue engineering on the breast because you are having the tissue which is changing all the time.
Mm-hmm.
Before the puberty, during the puberty, during the pregnancy, and on menopause. So each time you have different receptors, different function, and you would like to have harmonized all of these, otherwise you are getting problem, even cancer. So by using special technology called organoids,
Mm-hmm.
you are able to produce or reassemble the tissue that you're able to test your models, including pharmacogenomics, some other drugs, and so on. So we are moving step by step, but I am happy to tell you that in many institutions that I'm working, we are trying to synchronize our activities. We are trying to put all our brains together in order to answer the questions that we are not able to do. And that's one of the reasons why International Society of Applied Biological Sciences that I co-founded 29 years ago, why we are having this biannual meeting, having basically leaders in the field where we are discussing which direction to go, how we can unite the sources, knowledge. And for example, next year we are having topics which you are amazing. Basically, we are going on the roles. We are going to talk about roles of artificial intelligence and personalized therapies. We are going to talk about artificial intelligence driven diagnostics and treatment planning in regenerative medicine and the cancer treatment. But also, what makes me very, very happy, we are going to have 4 Nobel Prize laureates who are going to join us. So far, we have 10 of those outstanding human beings, and they are surrounded with the students, with the new generation of the top scientists. They are discussing, they are motivating, and one of the topics is going to be also intelligent machines,
Mm.
which Jeffrey Hinton said, some time ago, we are going to have systems capable of reasoning, learning, perception, communication. And he said, as you know, nobody has experience what will be happening if these things realize they are smarter than us. One issue is the medicine, and all other aspects of our lives is something else. So basically, that is what we're going to discuss, and certainly one of the most important topics we are going to have in Dubrovnik next year, in the June 15th to June 19th, will be artificial intelligence and multiomics, in order to understand everything what we discussed today, because I strongly believe that so many answers that we are not able to get today really rely what's happening on the level of the cells, including what you said, the gene therapy, CRISPR-Cas technology, eventually replacement of the mutation that eventually going to heal the patient. Because if you are talking, and we are coming back at the beginning about osteogenesis imperfecta. So if you have disease where mutation for collagen type 1 exists in every single cell, the only way how you're able to treat is to correct it. And to have the ratio of what we call healthy protein above certain level
Mm-hmm.
without changes of the mutation, which is going to lead to the damaged protein, we are not able to do it. So even during the period of my postdoc, we were able to do certain changes on the gene level.
Mm-hmm.
We did the cell culture, we did in the laboratory, but unfortunately, we never did on the patient. And that's also which make me frustrated for some period of time, but also put me in a position of the new challenges.
Absolutely. Absolutely. And that leads into one sort of final basket I want to touch on with you today, because I think it's extremely important, again, with where we are in 2025, as you're talking, globalization of medical training, advanced technologies, but at the same time, at least looking at the numbers here in the United States, a very low percent of the population that even seeks out clinical trials and innovative therapies when, they're sort of in that zone of no medical options available. Obviously, the medical tourism space in recent years has taken off significantly, and is a more structured system than it was, say, 10 years ago. Croatia seems to be coming online, and I'd love for you to talk a little bit about sort of your vision for where Croatia sits in sort of the medical tourism sphere, and then anything going on at the St. Catherine's Group and other organizations that you're involved with on that front, because I think it's very exciting in terms of the ability to sort of bridge these gaps across countries now and access these therapies wherever we want to access them.
You're right. You're right. Absolutely. So, as I said, for any individual, for any country, there is a old say which I strongly believe can apply to everyday philosophy of each of us, which says no wind can help the one who doesn't know where he's sailing. So I would like to see Croatia sailing stably, and without having knowledge-based society, this is not possible. Croatia is having very, very, very strong ties with the United States of America in many, many ways. And I see huge opportunity. Croatia is a member of the EU, having strong partnership with many countries, including Israel, including India. But we have to learn with one concept, which I strongly believe it's living with us when we are talking about wisdom, about philosophy for where we are going. And I said many times that wisdom is not knowing all the answers, but living in the peace with the questions, which is exactly what I'm thinking right now. So if you're talking about medical tourism, patient will come to you if you are able to provide excellent diagnostics, if you're able to provide excellent care, treatment, and rehabilitation. And they would like to see all at one place. But in the same time, to be very competitive, you have to have some additional knowledge, because if you are average, nobody would like to travel from the New York, Los Angeles, Tel Aviv, New Delhi to Croatia for a treatment. And that's why everything what I said, it's so important, especially translational medicine. So everything what we have today in the research eventually will be published, and some of those data will be transferred to the clinical work, which we call translational medicine. And I would like to see more clinical trials in Croatia. I would like to see more things going on in the field of the personalized medicine, and we are pushing strongly. We've been pioneering so many things, from including pharmacogenomics in the routine clinical base, in applying MSCs in a routine treatment. But at the same time, right now, I'm having so many patients coming for one, and we didn't touch this point, the one new field which is coming called Nutrigenomics. So it's a part of the personalized medicine where we are able to tailor dietary recommendation based on individual genetic profile. It's a completely new field where we are studying the interaction between genes and nutrition, enable to develop individual dietary plan. Once again, it's so important to understand what is happening with the metabolism of the lipids, of the proteins, of the carbohydrates. So we are able to do health optimization, disease prevention, and improve quality of the life. So as we speak, at St. Catherine Hospital, patient is able to come. We can do all genome sequencing. We are able to do multi-omics, including glycan analysis. And as you know, right now, it's very popular to compare chronological and biological age by analyzing glycans. We are able to provide nutrigenomics, pharmacogenomics, the state-of-the-art treatment from orthopedics, cardiology, spine surgery, rehabilitation, and in partnership with UPMC, indeed, on the cancer. But at the same time, we are not stopping. As we speak, in my institution, I'm mentoring 8 MD PhD students. And each of them, combining that work in Croatia and outside of Croatia, partnership with different institutions, and I would like to thank Mayo Clinic for being partnered with me so many years, and Professor Stanimir Vuk-Pavlovic from the Mayo Clinic, just bringing this ICEPS conference to Croatia. It's changing environment. It's making Croatia so popular. And then you are able to combine the beauties of the country. 1,000 miles of beach, more than 1,000 islands, incredible culture. My city, Split, is having one of the largest and oldest preserved palaces where people are still living. It's the Diocletian Palace. Zagreb, which is an amazing capital, great location, very close to Vienna, to Milan, to the Budapest, and so on. But without knowledge-based society, all these natural beauties are just one small portion of the entire picture.
Mm-hmm.
And you know, as Eleanor Roosevelt said once, and I love what she said. She said the future belongs to those who believe in the beauty of their dreams.
Yeah.
And I would like to every single person in the States, in Croatia, doesn't matter where they live, to have big dreams. And if you have a big dream, if I have a dream, our dreams will combine and we will change the world forever to become a better place for all of us. That's the society that I strongly believe, because what we are witnessing today, it's very, very disturbing. And I think that the role the scientists are having, nobody can replace, and that's my mission. And I'll do my best to live, to make a difference. I'll do my best to help anyone I can help. And indeed, I love enjoying living between 3 continents, meeting my friends, entering very deep in every single subject that we discuss. And I'll do my best not to get tired of doing what is good for the humans.
Well, you seem to have perpetual energy, and so it's extremely impressive. And I'm glad you brought up that point about nutrigenomics, because that's I'm a big proponent of. I spent a couple careers ago in the phytochemical space, and I'm a big proponent of using current technologies and to learn about everything we eat and all the phytochemicals that salad or these vegetables over here. I think we only scratched the surface. That's kind of cool that you're thinking about that. Dragan, one more topic I wanted to ask you about while we have you today, and again, it speaks to sort of the diversity of the research that you've been involved in. But I think is obviously extremely important part of your story is the work you've done on, I guess, sort of what we'll call the out of Africa hypothesis and the origin of early man. I know you've been involved in 2 major papers, I think, in both Nature as well as Science that discuss sort of the connection here and obviously the use of modern genomics technologies. Take us down this path as well, because I think it's a very important part of your scientific journey over the years.
Thank you, Ira. You're right. This science paper was pioneering analysis of the Y chromosome in order to understand where the European man is coming from. And our research helped identify distinct genetic lineage and their geographic distribution across the entire Europe. And actually, it supported the theory that modern Europeans descend from multiple ancestral population, including Paleolithic hunter-gathering and Neolithic farmers. And by using analysis of Y chromosome markers, it allows us for tracing paternal ancestry and understanding male-mediated gene flow over millennia. On the other hand, the paper that you're mentioning, Nature paper that came on the 2017, provided, in our opinion, groundbreaking insight into early expansion of anatomically modern humans out of Africa.
Yeah.
Actually, we studied 148 human population worldwide. And what was amazing, that the first migration, which came approximately 120,000 years ago, which was largely extinct and did not contribute significantly to genetic makeup of the present day non-African population. However, small group of anatomically modern humans left Africa and reached parts of the Middle East and possible Asia from that group that came 120,000 years ago. But what is important, in my opinion, to discuss that this early wave was followed by later more successful migration, which approximately happened 75,000 years ago, which became the dominant source of ancestry for modern non-African population. And these migrants spread rapidly across Eurasia, eventually reaching Australia and America. And I strongly believe that human migration out of the Africa was not a single event, but a complex process involving multiple waves, interactions, and outcomes. And I'm so grateful to all authors on these two outstanding papers, and it was a privilege to be one of these authors. And I hope that we help understanding and explaining in more detail than what we have before the history of the humankind.
Yeah, it's again another a very impressive piece of work, and it goes back to what you were saying about the merging of modern technologies to solve mysteries of us as people. So really, very impressive part of your story there as well. Dragan, I really appreciate your time. One last thing I just want to touch on, because we spent a lot of time on science and education, on medicine. We didn't say much about sport, but as you know, at the beginning, you are a recognized Taekwondo champ. I couldn't help but avoid your 2013 paper that you did with the folks here at Penn State and the University of Split and the Ministry on different fitness requirements for Taekwondo success. I didn't realize that Taekwondo was practiced on 70 different countries now all around the world. But what's happening in sport right now? And are you still competing? Do you get to match as much? What's happening?
Well, it's amazing. So basically, I was competing, and I was former vice champion of the former country. So basically, Taekwondo, it's my love, and so grateful for Taekwondo Hall of Fame in the United States for including me in the Hall of Fame in 2015. But today, I try to practice as much as I can. Indeed, I'm not competing, but I'm a member of I was member of the Council of Croatian Olympic Committee, so all time very close to sport. I was advisor to the president of the Croatian Football Federation and still senior advisor for him. And as you know, Croatia make remarkable progress, being second and the third in the war. And, I am on the second term member of European Olympic Committee Medical and Anti-Doping Commission, working all the time with the leading athletes, and currently vice president of the Croatian Taekwondo Federation. And most recently, I was appointed to be president of the medical committee of the Croatian Volleyball Federation, and became also a member of the medical committee of International Volleyball Federation, which always put me in position that sport is somehow my priority. And I'll just tell you something very interesting. During my term as a minister, in the period of 6 years, we were able to build, believe or not, 211 sport halls for the kids to practice sport in a small Croatia. And the sport is, it's been always part of my life, and it will be. Every time when I'm having a lot of stress, I'm running with the friends. Actually, it was a charity run yesterday, 5K, in the lake very close to here for the kids with a rare disease. And every time when we are having something like that, I'm with those who need it. And in the future, I strongly believe that sport will be making all of us much more united than we are today, because I'm witnessing what is happening on International Olympic Committee. All these efforts, people trying to bring through the sport, to bring people on the table to discuss more about the future, because sport is wonderful. You meet so many friends, you meet so many outstanding people that otherwise you would not meet. I just came back from Skopje, where we have a youth Olympic, we call youth European Olympic Games. Such incredible kids from the high school competing, discussing about future, discussing about exchanging training conditions. So that's something which make me incredibly happy, because what I witnessed today, the sport is is doing incredibly good. And at the same time, sport is able to put people on the table that otherwise they would never sit together.
Mm-hmm.
And I'll do my best, certainly, on that area as well.
No, it's again, yeah, very important message, and again, I love the sort of the interdisciplinary nature of all that we're talking about in sport and exercise. I think again fits very nicely in, as you're mentioning, sort of aging and biologic age, and I think we're as we continue to follow and understand, I think we'll have a greater appreciation for how much exercise it contributes to our longevity and healthy aging. So, a very important point.
And still, as you said, Ira, still so many things we don't understand. I remember
Right.
on early 90s when Henry Lee told me, as a leading forensic scientist in the States, he told me absence of evidence is not evidence of absence.
Yeah.
So today, what we don't understand, it doesn't mean that somebody in a few years will not understand. So I think it's so important that we keep going in the direction that we discussed today, that we keep people motivating. Otherwise, we will not be able to answer all these challenges which are, ahead of us. And so many time, I'm talking about how important it is that we help humankind. And I remember
Right.
what Ralph Waldo Emerson said, and I'm sure you're going to love it, because once in a while I'm reminded that I should be a better person. Because what he said, when you were born, you were crying, and everyone else was smiling. Live your life so that at the end you are the only one who is smiling, and everyone else is crying because you've been such a good human being.
There you go. There you go. Yeah. This is I really enjoy this conversation, and hearing so much about you, Dragan, and where you're taking this amazing body of work that you've been doing over the decades forward now. Any other final messages while we have you today, and then other public facing initiatives that concern you in terms of the remainder of 2025 as we get into 2026, whether that's conferences, talks, other public initiatives where we may run into and meet you on the circuit somewhere, please. And anything I didn't touch on that you want to mention while I have you today.
Thank you so much. Thank you. I'm so proud. I'm so proud to announce today, because it means a lot for so many people. The next ICEP's Mayo Clinic conference that we are going to have next year, June 15, June 19, 2026 in Dubrovnik, Croatia, where we are going to have the leading world experts in the field of artificial intelligence and the medicine,
Nice.
including 4 Nobel Prize laureates. We'll be discussing actually how artificial intelligence is changing the medicine entirely, from the cell level in the radiology, cardiology, orthopedic surgery, urology. Why artificial intelligence is going to be, I would say, the completely new concept that we have to introduce in the medical school as a subject. Otherwise, the physicians are not going to be competitive as we want to be.
Mm-hmm.
We are going to cover the topics, actually, including forensics. We'll be covering DNA-based identification of missing persons by using artificial intelligence. Artificial intelligence and forensic application in the justice system. Artificial intelligence-driven diagnostics and treatment planning, especially related to the cancer. So we have huge power in our hands. And the next year conference in Dubrovnik for 5 days will be focusing actually on the future, in which direction where we are going. Some of the most remarkable men live today in the medicine and field artificial intelligence. Will be there. About 700 to 800 participants will be joining us. And by the way, since we've been established, more than 7,000 participants came and they've been part of this conference. And traditionally, we are having one unit which makes me so happy. It's called Nobel Spirit. And it's a discussion, and it will be the 4th one, discussion between Nobel Prize laureates, students, myself, about in which direction the world is going. We'll be addressing different social, global, scientific challenges. We'll be discussing, I would say, brain drain issues and how science can change the world to be a better place. So usually we are picking up different locations by the sea, by the forest, sitting surrounded with the students and having very much open-minded discussion with so many questions that every time after Nobel Spirit session, I'm feeling thrilled because I didn't realize till that moment how many questions exist.
Right.
And some of the brightest kids coming from all around the world are coming to spend 5 days with such incredible community, and they're bringing the knowledge back home and sharing that knowledge. Usually, students and scientists from more than 80 countries are coming. And dear Ira, if there's any chance, I would like to have you as our guest. I think it'll be outstanding opportunity to have you with us, to meet these wonderful people, so that we can discuss jointly in which direction the world, science, technology will be going.
An extremely important discussion for an extremely important moment in time, and yeah, I'd be honored to be invited. But I mean, Dragan, this is such an exciting discussion. It's been amazing listening to you and this multi-decade journey that you've been on, and I just again wish you and all your team the best with all these initiatives as you move them all forward. Again, for everybody out there that's going to be listening to this episode across the podcast networks or who will be watching on the YouTube channel, again, you've been spending time with Dr. Dragan Primorac, amazing physician scientist at the intersection of personalized medicine, regenerative therapy, forensic genetics, Taekwondo, AI medical school, Nutrigenomics. I could list on and on, but Dragan, I want to thank you again for taking the time out of your schedule to take us through this amazing story. Obviously, thank you for everything you're doing to promote science and technology and move society forward. And as we like to say on our show, thanks for helping to create a better tomorrow for all of us via what you do. Really a great story.
Thank you so much, dear Ira. It was a privilege to be part of this wonderful show, and please count on me in the future.
Definitely. Great having you.